Thank you, Chuck, for this introduction. Welcome everyone on Zoom and in person. It's really a pleasure to be here in Cambridge. I've been attending a program at the Newton Institute on bridging non-equilibrium physics and biology. And, I'm delighted also to visit the materials science department on this trip. So, I'm here the rest of this week. So, if any of you want to catch me later for additional details, I'm happy to chat in person or later via email or on Zoom. So before I discuss the topic of the day that I think will be of interest for designing materials, I just want to give you a brief overview of the different kinds of things that we do in my group. So during my postdoc with David Nelson at Harvard, I was interested in knowing how thermal fluctuations affect statistical mechanics of thin sheets. And what was actually well known for the last 30 years or so, that thermal fluctuations make these sheets effectively stiffer for bending but softer for stretching. And you can get some intuition about these phenomena also if you play with crumpled paper. Just compare the how hard it is to bend the crumpled paper versus the flat paper, and you'll be able to see some of these effects. In the last few years, we also extended some of these results to try to understand what is the role of curvature. For example, if you have spherical shells or nanotubes. Interestingly, we found that because the mechanics of the shells is very different than mechanics of flat plates, we found that thermal fluctuations in spherical shells generate an effective compressive stress, which is entropic in nature. And what happens that if you crank up the temperature high enough, this compressive stress becomes so large that the shells become unstable, even in the absence of any external load, such as pressure, right? So they'll just become unstable on their own. And with Robin Bruinsma at UCLA, we did some study of the viral capsids, which are also small spherical shells. And interestingly, we find that some large bacteriophages, seem to be in a regime that we would predict to be unstable, and how they, deal with these to try to overcome these, dangerous situation. They basically self-assemble their capsids on scaffolds to try to, stabilize them. And once the capsids are fully assembled, they undergo maturation process, which makes these shells much more rigid and much more stable. While I was at Harvard, I also started interacting with engineers, in particular with Katja Bertoldi, when we were interested in so-called mechanical metamaterials, where the idea is that by designing the geometry of these structures, you can get unusual mechanical behavior. So here's an example of a structure which is a square array of holes. And when you compress these vertically, you will see that it will, buckle and also collapse horizontally. So it has effectively a negative Poisson's ratio, which is quite unusual for most ordinary materials. But here just a structural property, right? So this is just one of the examples, but in the last few years, there's been a number of examples of how people try to exploit these buckling instabilities for various functionalities. In my group, what we're interested in is how we can manipulate elastic wave propagation. So in particular, these are phononic crystals. So we basically calculate phononic band structures. And where are the band gaps or crossings, the degeneracies in these band diagrams crucially depends on the material property, but also on the geometric properties and symmetries of these crystals. But if you focus on this structure, once you will compress it, you will see that once it buckles, it drastically changes the symmetries, right? So basically, upon buckling, because you're going from one crystallographic group to another crystallographic group, this will lead to the degeneracies diagrams are allowed or not. So, we're basically exploiting, these symmetry changes to try to, mechanically tune these phononic band diagrams. In terms of mechanical metamaterials, we have also studied a lot these wrinkling instability. Basically, the wrinkling happens when you have compressed skin on a soft substrate, and you can try this with your hands, right? If you compress your skin, you will see that if you compress it enough, it will wrinkle, right? And we've, together with my colleague Howard Stone, we've also studied, what happens to these wrinkled structures when they interact with liquid droplets and so on, and you can get interesting behavior. But since coming to Princeton, I've also, expanded into a lot of new directions. In one of the very active area of research, my group was like the role of mechanics in morphogenesis of developing tissues and organs. So for example, with Bonnie Bastler, Halbert Stone, and Ned Wingren, we've studied the development of bacterial biofilms. And you see these like interesting patterns, and they come from the same wrinkling instability that I was just talking about on the skin when you grow them on a soft substrate. Together with Celeste Nelson, we've also studied the lung development, like how our lungs become all branched and how mechanics helps guide the branching process. With Daniel Cohen, my colleague, we've studied the expansion of like cell monolayers. So I'm going to show you a movie on these tessellated tissue, where basically like, we have like, they were seated in a stencil, which has this shape. And they're all identical cell types. These like red, blue, and pink, they're all identical cell types. We just use different dyes. And then we remove the stencils. And these cell monolayers will then start crawling and expanding because they want to, you can think of this as like a wound healing experiment. And, like what you see now when we remove the stencil, they expand and then, effectively like merge into one solid tissue. But you see that we have like this like very interesting artistic pattern inspired by the Escher. Right, so obviously these didn't happen by accident. We actually designed these tissues that, as these tissues expand, they follow like two very simple rules, right? So if I go back to the beginning, like, you will see that like, each edge, like cells at the edge of the tissue, they expand normally with a constant velocity. And this constant velocity is uniform and it doesn't depend on the local curvature. In particular, it's going to be the same in this nearly flat region and this highly curved region. So this is the same feature that we know from optics. The light front moves with a constant velocity, right? So we can just use the Huygens principle to try to see like how, the shape front of these expanding tissues evolves over time. And the other feature that you will see is that once these tissues collide, they just stop. They basically freeze, right? And so now I put this movie, you see that they just collide and stop. So if we just use these two simple rules, we can basically develop like a predictive computational algorithm that tells us how these multi-tissues will evolve over time. So, here the dynamic was particularly simple because, all tissues were identical. We can also play a game where we put like different tissue types, and then now they will expand with different velocities. And also once they collide, they don't necessarily merge. One tissue can, push around the other one and so on. So you can get quite interesting dynamics if you start playing this with multi-tissues. We've also studied the rheology of these so-called like vertex models of epithelial tissues, which these are like single layer cells that can often be well characterized with these like polygonal shapes. And the degrees of freedom are just the vertices of these polygons. That's where the name comes from. And we've tried to like systematically coarse grain and develop methods to see like like how we can start with these like mesoscopic coarse grain model to figure out like like what are the effective rheological properties on a continuum scale. And together with my colleague Sujit Dara, we've also been looking at the synthetic morphogenesis. So like now instead of working with like biological tissues, we work with like swelling hydrogels that swell around the obstacles to see how like as they swell, how they conform around these obstacles. And interestingly, what we found is that depending on the geometry of these obstacles, that there are some regimes when these hydrogels can just tear themselves apart, which is kind of surprising because you would think that these gels would be under compression once they hit the obstacles, but because they have to conform, like stretch around these obstacles, that there are like some areas that are under tension, and this tension can become large enough for the gel to fracture and tear itself apart. So we are also like very active in the studies of the phase separation of multi-component systems, which I'll talk about today. And this was actually also inspired by biological systems, in particular these biological condensers that I'll mention more about. And the last thing I'll mention here is that like many of the tools we developed for these projects will also be now combined in a new collaborative project on sustainable manufacturing and architecture that's led by Xu Yang at UPenn, where the idea is that we want to create like large scale, like self-folding origami and kirigami structures for the potential use in architecture, where we want to build, design this, make these novel materials from eco-friendly materials, where where we hope to also exploit like these phase separation ideas to develop like these multi-phase inks that will then like stimulate and actuate our structures, but we're also going to have to figure out how to overcome gravity on a large scale, right? Because in the last few years we've seen like a lot of development in the soft robotics in these areas, but like these soft materials won't really work if you try to go to the large scale. So this is like a brief overview, and now in the rest 20 minutes or so, I want to just tell you like something about the phase separation, how we can design these multi-phase structures. And like I said, idea came from the biology, and if you look at the classical textbook of eukaryotic cells, you will see that it has like a lot of internal structure, and often like each of these like internal organelles are drawn like with its own membranes, and for many years biologists thought that that's how the cell is compartmentalized and kind of divided. But in the last 15 years or so, it was recognized that there are also some organelles that are membraneless, and they form via liquid-liquid like phase separation. Just like when you mix oil and water, they hit each other, so they will phase separate, right? And like these biological liquid condensates are implicated with a lot of different biological functions. So they can organize genome, they can sequester unwanted materials, like they can act as like mini factories and so on. And here are just like some recent review papers that describe like many of these functions. But often like these droplets, once they form, they're not just like simple like liquid droplets, but they also have internal structure, which is related to the functions. And just to give you an example, I'm going to show you this hierarchical structure of the so-called nucleoli, which are in the cell nucleus, which are basically used to speed up the assembly of the ribosomes, which are also very important for biological functions. And here, like you see, is like these are like fluorescently labeled molecules, like like each color fluorescent labels different molecules, which are enriched in different phases. And you see these like most inner phase is the blue phase, which will like form around specific regions on the DNA that encode for the ribosomal RNA. And then they'll go to the next green phase. Well, this ribosomal RNA will be processed. And finally, in the red phase, this ribosomal RNA will then self-assemble together with other proteins into a functional ribosome, and then functional ribosomes will then be exported out of the nucleus. So you see that this hierarchical structure kind of guides this reaction and assembly process, and tries to facilitate this, right? And here is like an experimental image of these nucleoli in the frog egg cells. And once you disrupt the cytoskeleton, you will see that these droplets will just like merge and coalesce. And if you're just looking at this video, this looks just like ordinary fluids, right? But in fact, these are like complex fluids because they're made of like many different components. Like our cell has lots of different proteins and molecules that enter there, right? So these are really complex fluids. So like these kind of made us think about how we can think about the phase separation in these like multi-component settings. Another thing I should mention, like often when you think about cells, they're very active, they burn energy and so on. So they're usually like out of equilibrium, right? But for now today, like, but it is believed that like this phase separation process is really driven by the classical thermodynamic forces. So for now, I will, strip off all the non-equilibrium effects and activity in cells, and I'm just going to focus on the phase separation in passive multi-component mixtures, right? And here, just an illustration of the things we want to, investigate. So let's say that I have a multi-component liquid mixtures made of different molecules. And, what I would like to answer is, if you tell me, what concentration different molecules you have and how they interact, can I predict how many different phases will form? what will be their composition. For example, the blue phase may be enriched in star molecules and the red phase may be enriched in circle molecules, and also how they will arrange in space. Okay. Okay. So first, I'll just in 2 slides kind of mention like an algorithm that we developed for constructing phase diagrams by convexifying the free energy. And this algorithm was inspired by Carlos Marques and Fabrice Talman's group in this PRL paper from 2011, which was designed for ternary systems, but I'll tell you how we generalize this to more than 3 components, right? And the idea is the following. So, in material science, it is well known that, in equilibrium, the system has to lie on the convex hull of the free energy, right? So, like if the free energy is non-convex, you have to basically convexify it first, right? And basically, I'm going to tell you how we can basically construct a convex hull of the free energy and how we can read off the phase diagram directly out of that information, right? So here is just going to be illustration for the ternary system, and then I'm going to comment how to generalize this to the more than 3 components, right? So in the bottom left corner, you see like a phase space of all the possible mixtures, right? So, like here at the, like we can start basically, as I go from different points in this space, like I can basically like vary concentration of molecule C3 by going from the bottom to top, or like, I can vary concentration of C1 by going from left to the bottom right, and so on, right? So each different point in this triangle will describe like a different possible mixture, right? So then the idea is, like I will discretize this phase space of all the possible mixtures. And then for each of these discrete points, I need to evaluate the free energy of that mixture, right? And, if I have a model, I will just calculate it, or, I can run simulations, or I can measure this in experiments. So just try to get a value of the free energy of each of these possible mixtures. So now I have a discrete approximation of my free energy surface in this phase space of possible mixtures. And then we just convexify this free energy by using the standard convex hull algorithm in computer science. And when you project this convexified free energy back to the phase space, you will see that some of these triangles remain small, similar to what they were at the beginning, but some of them merged and got stretched. And these like merged and stretched triangles, these are signatures of the regions where the phase separation happens, right? And what was the novelty of these PRL paper from Carlos Marques and Fabrice Talman? They figure out how to interpret these stretched triangles, right? So in particular, they figure out that if the triangles got stretched in one direction, this would correspond to the 2-phase regions. But if they got stretched in 2 directions, this would correspond to the 3-phase region, right? And how do we know whether they got stretched or not? Basically, we look at the Euclidean distance between these points in this phase space. And if this Euclidean distance is comparable to the initial mesh size, they basically correspond to the same phases, right? So these 2 are close, these basically correspond to the same phase, but these 2 are far apart, so they correspond to different phases, right? And whereas like here in this like sample on the right, like all the distances are much larger than the initial mesh size, so they would correspond to the 3-phase regions, right? So once we have this information, we can just color these triangles, and now we have a phase diagram, right? So now if you tell me, your initial concentration of the mixture, basically I can, find it like where it is in these convexified phase space, like in this projected phase space once we convexify the free energy. And basically then we know that the compositions of the coexisting phases will be at the corners of these points. So for example, like in these regions we'll have 2 phases. One phase will be at the top corner, one will be the bottom corner. These are also known as the tie line in the phase diagram systems. But if you start in this region, we'll have like a triangle that corresponds to the 3 phases. The compositions of the 3 phases will be these 3 corners, right? So we know that the compositions, we can also figure out, what will be the relative volume fractions of each of the coexisting phases by just using the usual lever rule, which will, make sure that the average concentration is the one that we mixed in the closed system. And furthermore, like we can also estimate the surface energies between the pairs of the coexisting phases if we know what is our free energy surface, like once we know the compositions of the 2 phases, right? So, like these concepts can easily be generalized to more than 3 components, right? So, like now, for example, if you have like a 4 components, then the phase space would now be like a tetrahedron, or in general, like, like would be just like a simplex, in n dimensions if you have like n components, right? So then we always like discretize our simplex. We generate like a free energy surface, discretized free energy surface by evaluating the free energy for each of the possible mixtures. We convexify it, and then we project it back to the phase space, and then we just have to, look at our, now deformed simplices, and figure out, like by just looking at the Euclidean distance of, different corner points of the simplices, to figure out whether the corner points correspond to the same phase if they are close, or to the different phases if they are apart, right? And like where the discretization becomes important is, like if like you want to have like if you want to resolve the phase boundaries accurately, then you and that becomes especially important like near the critical points, because then the compositions of the coexisting phases are like very close to each other and so on. Okay, so this is like just like a quick overview, and you can find more details in the soft matter paper, or I can answer some questions at the end of the presentation. But now I want to focus on how these multiple phases, how they arrange in space. And here, just for illustration purposes, I'm going to use the Cahn-Hilliard simulations of the Flory-Huggins model of regular mixtures. And this is just for illustration. Everything I'll tell you is very general and can be applied to any other model, right? So here, in the Flory-Huggins model, the free energy density per volume, normalized by Katie, has 3 contributions. One is from the mixing entropy, these log terms, where Ci are just the concentrations of different molecules. The second term describes their relative interactions, where chi-ij is just the matrix encoding these interactions. And the last gradient term will capture the interfacial properties of the system, where the lambda is the characteristic like width of these diffuse interface. In the Cahn-Hilliard dynamics, here this term is the chemical potential. The gradient of chemical potential will drive flow. So here we use the Kramer's mobilities, which depend on concentrations. And for simplicity, well, I'm just going to assume that all the small molecules have the same diffusivity, and then divergence of the flow of the current will tell me how concentrations locally evolve. You'll notice that in general we also have like a thermal noise, but here we'll not do that, and we're just going to focus on the spinel decomposition. And I'm going to make like a 3D simulations with periodic boundaries, where I'm going to just tune the concentrations and their interaction parameters, chi-ij. And like now, here are some representative simulations, and you can see that we can generate quite different morphologies if we tune concentrations and interaction parameters. You also notice that these structures like grow and coarsen over time. This is known as Ostwald ripening, and it's well known that like these structures, they grow with the characteristic size grows as a time to the 1/3 power. So we wanted to understand, how we can control these morphologies, and everything I'll tell you is based on this PRL paper that we published like a few years ago. Okay, so what control, and also like I'll mention, which is also known, seen here from these equations, that we're going to assume that these droplets are small enough that the diffusive and interfacial effects are dominating. So we're going to ignore any hydrodynamic flows, advective flows, and we're also going to ignore any buoyancy effects if there's like a density mismatch between phases. Okay, so let's start with 3 fluids phases. And in fluid mechanics, is well known that depending on the values of the surface energies, you can get either partial wetting or complete wetting. And partial wetting you're going to get when each surface tension is smaller than the sum of the other two. In this case, like you're going to form stable triple junctions, where basically you have like a force balance between different surface tensions, and these force balance will also determine the contact angles between these phases, right? And when you run simulations, you'll see these triple junctions are very stable and they're quite long-lived. On the other hand, if one surface tension is larger than the sum of the other two, then these small surface tensions can never balance this large one. This means that these triple junctions, if they form, they're mechanically unstable. And this means that the red and blue surface tension is going to pull this green phase in between, and to try to completely eliminate the interface between red and blue to try to get rid of this large surface energy, right? And when you run simulations, you see that now these like red and blue blobs are completely shielded from each other. There's always like a layer of the green blob in between, right? So now we want to figure out how to generalize these wetting conditions to more than 3 fluid phases, right? And here it will be convenient to introduce this graph approach, where each node corresponds to a phase. And if the 2 phases share an interface in real space, we're going to make a link in a graph, right? So for the partial wetting case, like each phase is in contact with every other phase. So we have a fully connected graph. But for the complete wetting case, red and blue are not in contact with each other. So we have a missing link, but they're still in contact with green, right? And every time you're going to see these graphs, we can also then invert them to the inequalities for surface tangents if needed. I'm also going to comment on the volume fractions. So here in these simulations on this slide, I had equal volume fractions of all the 3 phases. If I increase the volume fraction of the blue, which is now semi-transparent, and reduce the volume fraction of red and green, then red and green will break into small droplets because of the Rayleigh-Plateau stability. But you see that these connectivity graphs will remain the same, because in the left, red and green are still in contact with blue. So we still have like all the phases are in contact with each other. And on the right, these like red droplets are encapsulated by the green phase. So they're still never in contact with blue, right? So this suggests that these connectivity graphs are completely determined by surface tension, whereas the geometry of the structure also depends on the volume fractions, right? So in the rest of the talk, I'll just focus on the facts of surface tensions, which determine this connectivity graph, which in some sense you can think of sort of like a topology of these structures. Okay, so now let's say that, through whatever method, we have determined that we have 4 coexisting phases, and we have already measured all the surface energies, right? So somehow we should be able to use this information to predict how they will arrange in space. Okay, and so we already know what happens when the subsets of 3 come together, because we know that we're going to get either partial wetting or complete wetting. So let's say in this example that the red, blue, and green partially wet each other, but the red one completely wets white and green, and the red one completely wets white and blue, and green, blue, and white partially wet each other. So now we can, we should be able to stitch these local rules to figure out what happens when all 4 of them are present. And the algorithm is very simple. We start with a fully connected graph. We iterate over subgraphs, and every time we see a missing link, we remove it in the main graph. So this predicts that the red one should isolate the white from the green and blue, which are still in contact with each other. And when you run simulations, that's what you see. You see these white blobs are always shielded by red, and they never get in contact with green and blue. All right, so the reason why this works is that like white does not want to be in contact with blue, because if they're in contact, you can always insert a little bit of red phase in between to lower the free energy of the system. For the same reason, white does not want to be in contact with green, because it can insert the red phase to lower the free energy. But all the other links will be there, because if the system is initially well mixed, it's going to transiently, all the phases are going to have a chance to interact with each other. You're going to form these like stable triple junctions where you have partial wetting conditions, which will be there unless they get pushed apart by these complete wetting condition case, right? And obviously, like, these are like equilibrium systems, but, like if you prepare a system which is not well mixed, you can also end up in metastable states and so on, right? I'll just mention that, we can get to the same final connectivity graphs in multiple different ways, and that will be useful when we want to do the inverse problem. For example, the only difference between the top and the bottom is that most wetting conditions are the same, but on the top, green, blue, and white partially wet each other, but at the bottom, green will completely wet white and blue. So basically, if you remember, this will make sure that the link between white and blue is removed in the final graph. But this link was already removed earlier because the red one also completely wets white and blue. So in the bottom case, we removed this link twice, but in the top case, we only removed it once. But the final morphology will be the same regardless. And you can see when you run simulations, they look very similar, right? But the transient structures, like on the way to the equilibrium structures, could be different, right? Because in the top, green, blue, and white will stuck together until like the red one pushes them apart later, whereas at the bottom, the green one will always like immediately push white and blue apart even before the red one comes along, right? Okay, another question you can ask, like if we now tune the surface tangents, like what are all the possible ways we can rearrange these phases? And it turns out this is just, we can enumerate these by just enumerating all the topologically different graphs. So here with 4 phases, there are like 6 possibilities, which you can see here. When you have 3 phases, there are only 2 possibilities, complete wetting and partial wetting, which I mentioned before. When you go to 5 phases, you may have 21 possibilities, and this goes exponentially when you start adding more and more phases. But the good thing is we can actually realize all of them. Okay, so finally, and you can see that these morphologies look very different, and all you need to do is change surface tangents to generate them. Okay, so finally, I also want to mention the inverse problem, right? So let's say that for whatever reason I want to make this structure. So the way, and now you would like to know what you need to mix together in order to generate that, right? So like first I'm going to create a connectivity graph that corresponds to this structure. Then I'm going to break into the subgraphs of all the possible triplets. And now I need to know how to, use this information to figure out what to mix together. So here you will recognize some graphs that look like complete wetting, some graphs that look like partial wetting, but also some graphs that are disconnected, and we don't really know what to do with them, right? So maybe we can just ignore them because I told you there are many ways how we can get to the same final connectivity graph, right? So let's see if this works, right? So this graph will make sure that the link between gray and red is eliminated. This graph will make sure that the link between gray and white is eliminated. These 2 graphs will make sure that the link between blue and green is eliminated, but none of these graphs will ensure that the link between gray and green is eliminated, right? So among these disconnected graphs, I have to impose some more constraints to make sure that the link between gray and green will be eliminated. So I'll assume that the red one completely weights green and gray, but I could have also assumed that the blue one completely weights gray and green, like it doesn't really matter, okay? So now what I do, I use these graphs and convert them to the sets of inequalities for surface tangents, right? And now in principle, I have infinite solutions for these inequalities or no solutions. And it turns out that here we always have infinite solutions. We can always find, a set of surface tangents that satisfy these inequalities. And then, like now we just need to convert these to the, chi parameters in our model. And, then we just tweak concentrations to get the right volume fractions. And here is, the structure that looks very similar to what we want. Of course, these are fluid, so they can easily be formed, so they don't look exactly like that. But, qualitatively, like they look very similar to what we wanted to design. And here are some other examples that, may be more practically relevant. So, like if you want to make like this like nested structure that I showed you initially from the biology, this just corresponds to the line graph. And maybe like in some applications, maybe you want to have like these like multi-phase emulsions that are encapsulated, which would just correspond to these different types of graphs. So, like now this graph approach, can be very elegant and can, like easily like help us like arrange these different possibilities. Here, I apply these to the liquid phases, but, I think it can also be generalized to other kinds of systems where, like where the interfacial energies are dominant, but, you can also try to, include like other types of energies. But, I think this graphical approach can just be like a neat way of organizing these. So now we're also trying to, like test these in experimental systems. So we partner with Omar Saleh's group in Santa Barbara, which is making these like liquid droplets made from like DNA strands. And, like by mixing different types of DNA strands, we can control the relative interactions between these strands, which can hopefully be used to tune these surface tangents. And, we're also trying to do some molecular dynamic simulations to try to guide the experiments. Finally, I also mentioned that, so far I only talked about the phase separation in liquid mixtures. Like in biology, these inside the cells will also have like cytoskeleton, other filamentous networks. So it's also interesting to explore, like how these condensers phase separate in the presence of these fibrous networks. And, here, like together with my colleagues, we just figure out, like what constructed the phase diagrams just using simple scaling arguments, and figure out that, there are 2 important non-dimensional numbers. One is the elastic Kepler number that captures the effect of the surface tension, the stiffness of the network with the shear modulus G, and also the characteristic pore size. So on the left, you're going to have very soft networks. On the right, you're going to have stiff networks. No, the other way on the left is going to be stiff network. On the right, the soft networks. And then on the other important non-dimensional parameter is this permeable elastic number, which tells you like how much the droplets like to wet the network compared to their stiffness. So basically, you see that if the droplets do not like to wet on the top, and if the network is stiff, then you're going to have these like micro droplets that are going to be constrained to the sizes of the pores of the network because they're not surface tension not strong enough to deform the network. But if the network becomes sufficiently soft, these like mesoscale droplets, and here eventually they will not coarsen indefinitely, but because many of these networks are strain stiffening, the more you deform them, the stiffer they become, which will typically select a characteristic size of the droplets. And that's also what you see in biological systems, that often these droplets form like a particular size. But if you lower this permeable elastic number, then like if the droplets like to wet the fibers, they can also encapsulate these fibers. And like if they really like to wet them, eventually they're just going to coat all these fibers. So this would be like another possible phase. And then the droplets also don't necessarily need to remain spherical because they can deform. So we've done some preliminary simulations related to this work, where we looked at the phase separation inside an random elastic networks. And in the top left corner, you can recognize this cavitation phase. In the bottom right, you can recognize these microdroplets where the network cannot be deformed. And in the bottom left, you have like this permeation where you see that these condensers also like encapsulate some of the fibers and deform this network. And finally, I'll just mention that, together with my colleague Cliff Brangwine, we're also testing some of these ideas inside the real cells. So Cliff Brangwine has designed this like an optogenetic correlate system, which can induce phase separation when you shine the laser light inside the cells. They have also engineered these correlate systems, these droplets, which can bind to the telomeres, which are part of the genome inside the cell nucleus. So now you can see that I'm going to show an experiment of how the induced droplet between these two telomeres when they shine the laser. So now you see the droplet is forming, but now when they turn the laser off, the droplet will dissolve, and these like capillary forces from the surface tensions are going to come together and merge these droplets. And we can also like simulate this process where we can induce the phase separation by just like locally quenching the system. Then we dissolve these droplets by removing the external stimulus, and we can bring this together. Like depending on the local environment and where how far apart they are, like there are also cases where the droplets, the telomeres don't necessarily merge, but once the droplet dissolves, actually the telomeres can detach and then go apart, so they can recoil. So we can see these also in simulations. And finally, like the local environment is not necessarily homogeneous; it can be heterogeneous. So if you look at the telomeres that's close to the cell the nucleus boundary and the interface, you see that the movement is asymmetric. In particular, the telomeres at the boundary are quite stuck, where the telomeres in the center can move a lot. So basically, like from this movement, you can also extract some properties about the local viscoelastic properties of the media. And we've also demonstrated this numerically. And like this is now shown in this preprint that's you can find on the bioRxiv. So with this, let me thank you for your attention. I will just briefly acknowledge, who are the main people that contributed here. So a lot of the work was done by my former postdoc, Sheng Mao, who's now assistant professor at Peking University. Milena was a former undergraduate at Princeton who came up with the idea of using graphs to kind of organize these multi-phase morphologies. And, that was the crucial insight that we needed to then, organize everything. And a lot of this work was also done with close collaboration with my colleague, Mikko Hataya. Pierre Vanstray was like a former postdoc at Princeton, who is now like a professor in France in Marseille, and also has a position at CNRS. And he did these like simple scaling arguments to figure out how like what happens when you have phase separation inside the elastic networks. Hongbo Zhao is like my current postdoc, which has done some of these simulations to test Pierre's predictions. And he's also been simulating these like optogenetic experiments that were done in Cliff Bergman's group. Most of the time I focused on this PRL paper to describing morphologies. I also briefly mentioned this like soft matter paper for constructing like phase diagrams using this convex Hall method. And again, I just briefly mentioned these two other papers. So with this, thank you for your attention, and I'm happy to take any questions.